is joining. I guess they thought it was over. Oh wait, there's a new person. I'll wait for just one more minute. Okay, I don't think anyone else is gonna come. Maybe they thought it was over. If they come in later, well, that'll just happen. Oh, I didn't start sharing my screen yet. I'll start that now. Wait a minute. Okay. Can you see that? Where's the chat? Can you see the screen? All right, that's great. Okay, so hello, my name is Brenda. I am a rising freshman from Michigan, and I will be teaching genetics and heredity. I wanted to teach genetics because in middle school, I was a part of the Science Olympiad team for 2 years, and I did heredity as an event, so that is why I am teaching. Hello, Zoe. This is my first time teaching a class like this, so if there's any feedback you have for me, like if you want me to go slower, go faster, please don't hesitate to ask me. You can feel free to use the Q&A feature to ask any questions or just type it in the chat. Please try not to, I guess, spam too much in the chat because it's currently open to all of everyone. So if it gets too disrupted, I'll just turn off the chat to make it only panelists. Anyway, let's get started. Okay. So this first class, we're going to talk about Mendels Laws. So first of all, what is genetics? Genetics is the study of heredity. And then you might be asking, what is heredity? Well, when you hear the word heredity, you might think of a word like inherit. It's kind of similar, I guess, or inherited. So heredity is just the passing down of traits from parent to offspring. So like people will often say like, oh my God, you have your mother's eyes or your father's nose or something like that. And it's because your parents pass down their traits to you. Oh yeah, I forgot I had that. So it's the passing on of traits from one generation to another. Okay, so at the beginning, I mentioned that we're going to be talking about Mendel's laws today. So Gregor Mendel is known as the father of genetics. He was an Austrian scientist from the 1800s. He experimented with pea plants. He formulated 3 laws, but unfortunately, nobody cared about his work until the 1900s, and I'll explain why later. But he is very important whenever you talk about genetics and heredity, and if you ever take a test on heredity or genetics, he will probably appear somewhere. So why pea plants? He decided to work with pea plants because they're easy to grow, they reproduce rapidly, and their traits are very easily observable. For example, something like my hair color or my skin color, you can easily see that. Like you can look at me and say, oh, your hair is black and your skin is like fairly pale. But something like my blood type is much harder to observe or like find out. You need to take a blood test. So for pea plants, there are many traits and they're all very easily observable, like plant height, flower color, flower position, seed color, seed shape, pod shape, and pod color. So these are the 7 things that Mendel observed in his experiments. So traits are the characteristics of an organism. You might have heard traits as in like character traits in English, maybe. Maybe you had a character, you need to analyze character traits. So those are just like the characteristics of and so within traits, there are 2 types, I guess, 2 categories, inherited traits and acquired traits. So inherited traits are the thing I talked about at the beginning, which are something the parent passes down to the offspring, like hair color, eye color, that kind of thing. But acquired traits are traits that are derived from environmental influence or outside influence. For example, if I'm in the sun for a long time, I'll get tanned, you know, and if I accidentally cut myself somewhere, I'll get a scar. Acquired traits can't be passed down. Like if you had a scar on your arm, I don't think you can't pass that to your child. Back in the, I actually don't remember what time period it was, but there was a time when there was this French scientist actually who thought that you could pass down acquired traits. The example in my textbook and from 6th grade was giraffes. So giraffes, they need to like reach up to eat leaves from trees, right? So he was like, because each time they stretch their neck a little more, you know, to reach up there. So every time they breed, their necks get longer. And that is false. And I'm pretty sure it was disproved by Darwin, I believe. Okay, so the 3 laws of that Mendel formulated are the law of dominance, the law of segregation, and the law of independent assortment. So today we're only going to be focusing on the law of dominance because with segregation and independent assortment, we're going to need to learn about like 3 more topics, and that's just too complicated for one day, especially because we're running late on time today. So I have a brief description of each law just because, just to understand that a bit better. So the law of dominance is that dominant alleles will mask recessive ones. And you'll learn more about what alleles are and like the difference between dominant and recessive in a later slide. The law of segregation is that each parent gives 1 allele. So you might have heard that you get 50% of your DNA from your mom and you get 50% of your DNA from your dad. And that is because each parent is only giving 1 allele. It'll make more sense later. And independent assortment is that different traits are inherited separately. This one's a bit easier to understand, I guess. So it'll just be like, my blood type is not related to my eye color, you know, like just because I have a certain blood type does not mean I will have a certain eye color. However, there is an exception to this because you will notice that, for example, in Asian people, we usually have dark hair and dark eyes, and that is because of linkage. But that is kind of complicated. It's just when the genes are kind of close to each other, and so they're more likely to be inherited together. They're not like actually inherited together, just like more likely, you know? That's kind of confusing, but don't worry about it. Okay, so vocabulary. Like I said earlier, everything I just said will make more sense. So a gene is a segment of DNA that codes for a specific trait. So we know that DNA is like the stuff inside of our cells, and that just like codes for everything. So a gene would just be like a specific segment of it, and in that segment, this is complicated. Okay, so that segment just codes for some specific trait. And alleles would be the 2 or more versions of a gene. For most of this lesson today, we're just going to be talking about 2 alleles because it will get complicated later on. But for now, there's 2. There's a dominant one and a recessive one. And dominant alleles are represented with a capital letter, and recessive alleles are represented with a lowercase letter. And I just have a simple definition right here, purebred, 2 of the same alleles. And that is because on the next slide, I'll be talking about purebred a little. I'll go more in depth on what it means later on, but it has to do with Mendel's experiments. Okay, so just as a question, what do you think happens when you breed a purebred tall plant with a purebred short plant? Remember, purebred means that they have 2 of the same alleles. It's a mostly rhetorical question, so you don't need to respond. But anyway, most times you would think that if you're breeding a tall plant with a short plant, you would get a medium-sized plant, but that is not true. So Mendel discovered that when you took a purebred short plant and a purebred tall plant, you can see, where's the annotated features? Okay, you can see here, this is the alleles for this tall plant, and these are the alleles for the short plant. When you breed these together, you actually get still a tall plant. And this is what the law of dominance means. You can see there's a dominant allele right here and a recessive one, but because there is a dominant one, it will mask the recessive one. So part of the reason that Mendel's work was so largely ignored and just like brushed off, and people kind of like scoffed at it, was because they didn't think it made sense. You know, back in the day, a lot of their reasons didn't make sense. You know, they thought everything revolved around the earth, that kind of thing. They were like, if you have two plants of differing heights, it doesn't make sense that they wouldn't mix. It doesn't make sense that you would still have a tall plant. But then it's such a nice word. Okay, so more about the law of dominance. Like I mentioned earlier, it just means that dominant alleles will mask recessive alleles. And as long as there is one dominant allele, the offspring will have the dominant trait, which is what I was annotating earlier. You can see that here, 2 dominant alleles, 2 short alleles. When you breed them together, 1 from each, right? Because you get 1 allele from each parent. Then you have 1 uppercase and 1 lowercase. However, just because there is 1 uppercase, it means you will have the dominant trait. However, because this plant has both of these alleles, it may come up later. Like if you were to breed this plant with, say, another plant, pure red short plant, why not? You might see that their offspring could be short again. And again, Mendel discovered this too, and people were like, okay, no, that doesn't make sense at all. So for some clarification, T is the uppercase letter, it is dominant, and it represents the tall allele. And lowercase t is the recessive one, and it represents the short allele. So now we have some definitions, which I have highlighted in green, but really it doesn't make it stand out. Anyway, so hybrid or heterozygous means 2 different alleles, which is what this would be right here, because there are 2 different cases of allele, like uppercase and then lowercase. And then purebred, which I mentioned earlier, and homozygous means that 2 identical alleles, which are both of these plants up here. Hybrid and heterozygous mean the same thing, and purebred and homozygous mean the same thing. In problems, you might see them interchanged, but they just mean the same thing. It really depends on who's writing the problem. I tend to use heterozygous and homozygous more often, but they mean the same thing. Any questions on this? I know it might have been a bit confusing because there's a lot of vocabulary involved. No? Okay, I'll take that as no questions. Anyway, next we have Punnett squares. So Punnett squares are very essential for genetics and heredity because they allow us to see possible. Oh wait, we have some chat. Oh, okay, you've learned this before. Very nice. Punnett squares are very valuable because they let us see the possibilities in offspring. So they're created by Reginald Punnett. He named them after himself, which is fairly obvious. But I have a empty, sample Punnett square over here, and you can see that it's basically like a multiplication table almost. You have the allele 1 of parent 1, allele 2 of parent 1, and then the same for parent 2 over here. And I'll give an example on the next slide, but when you find the possibilities, you basically just take this allele and put them together. I'll make more sense later. But before we move on, we have 2 more vocabulary words. Another chat. Okay. Another set of vocabulary, genotype and phenotype. Genotype, think gene at the beginning, is the genetic makeup and it's symbolized with letters. So it's the actual coding of the trait. So for example, these letters. If I say TTVT over again, it doesn't make sense. So just take a look. And phenotype, PH, physical, I guess. So phenotype are the physical characteristics. For example, tall, short, something that can be observed, like hair color. I come back to hair color a lot. But so remember, genotype is the genes. It's behind the phenotype, I guess. They create the phenotype. They code for the phenotype. Anyway, phenotype is just physical characteristics that you can see. Okay, so here we have an example. So this is the same Punnett. This would be the Punnett square for the example I was talking about with the law of dominance earlier. You can see I have the same picture here just for reference. But this is the tall plant, and this is the short plant. So we take these alleles and put them in separate boxes. So we have the first capital letter, capital T here, it's a capital T, We have 2 lowercase t's here. Put them in separate boxes. You might hear sometimes that you put like the father on the top and then the mother on the side, but it really does not matter. I don't know. I think I asked once in when I was in like a genetics class for 6th grade, but it doesn't matter. It really doesn't. And so now we can see that it's kind of like a multiplication table. So if you had a multiplication table that was something like, that is a really bad square, I'm sorry. And so we put the x here and we put like the 2 here and the 2 here, and you multiply them together, it's 4, you know, because it's 2 times 2. Kind of do the same thing here. We just put the t here and then put the other t right next to it. And yeah, all of their offsprings will be tall, but they will all be heterozygous or hybrid. If you remember, that means they have 2 different alleles. They will all be tall, but they'll all be carrying the short allele, so it might come up later. Okay, so that's just an example of a Punnett square. Oh wait, I forgot. I really forgot how I made this presentation. Yeah, so you can see we have the alleles of parent 1, and then the alleles of parent 2, and then the 4 in the center are all possible outcomes. Usually when drawing a Punnett square, we just like draw a grid, put the alleles on the sides, and then just like put them together. However, in Google Slides, it looks kind of awkward, so I just made an entire table. Okay, anyway. Some more Punnett squares. So these are the common ones, and that's because they're, I guess, very commonly used, maybe. It's because they're like the standard, you know. If you were ever taking a heredity test, they might ask you for like the ratio of this, like just from memory. It's because these are very common. I don't know how to explain it. These are just common. They're standard. So this first one up here, that's a bad arrow. This first one is called a monohybrid. So mono meaning 1, and hybrid meaning you have 2 parents and you're crossing them together. Kind of confusing, honestly, but oh well. And this is the Punnett square that I was using earlier. And if you're ever taking a heredity test, this is probably the most standard one you'd see, because most of the time it isn't accurate because genetics is so much more complicated. But for our purposes, this is the most common one. So you can see we have a heterozygous cross, which is the most common cross. For standard crosses, I really don't know how to put it. It's just like you cross heterozygotes. So we can see we have the capital letters and then a lowercase letter, capital, and then lowercase, and then we cross them all together. And the reason why these heterozygote crosses are so interesting is because you have all 3 possible genotypes, which is the genetic makeup, if you remember, which is the letters. So the phenotypic ratio would be 3 to 1. So let's just say the T gene represents height, and capital T is tall while lowercase t is short. So you can see that these 3 are all going to be tall, while this one over here is going to be short. I need to undo that. Ooh, okay. So that's a phenotypic ratio because you're just looking at the physical characteristic that this codes for. However, for the genotypic ratio, it's slightly different because we're taking a look at the actual genotypes, like the different combinations of letters. We have 1 homozygous dominant, not heterozygous, homozygous dominant, which means that is 2 capital. And then we have 2 heterozygous. You don't attach a dominant or recessive to heterozygous because it's automatically going to be dominant. And we have a homozygous recessive right here. And you can see the ratio is 1 to 2 to 1. This is a very common ratio, might come up in some tests. They might just ask you for like the ratio. And if you have it memorized, it's a lot easier than actually making it fast. And then we go on to dihybrids, which are painful to say the least. So di meaning 2, and hybrid, again, also meaning 2, because, oh, well, bye. I hope you learned something. So di meaning 2 and hybrid, again, because there are 2 parents. So this one is where it gets more complicated. And I'm not going to talk about these ever again in this presentation, but I'm just putting it here for reference because you will see them if you choose to go into like Science Olympiad and do heredity or something like that. So dihybrid means you're crossing two trees. So for example, this one, I kind of had the colors. I forgot to put like the sticks in because I did it earlier, but then I deleted it. Don't worry about it. The T gene still codes for height, with capital being tall and lowercase being short, while the Y gene codes for color, with yellow being dominant and green being recessive. Most of the time, in genetics problems, I guess, the dominant, you know what, it doesn't really matter. They just assign a letter to it. Sometimes it's like the first letter of the trait itself, but it really does not matter because you can just assign any letter. It doesn't matter. Okay, so this is where it gets slightly complicated. I'm just going to go into this as briefly as possible. So again, yes, Gregor Mendel, he was the father of genetics, you know? And okay, where was I? Yeah, so it's kind of the same thing. You can see that we still have like the alleles on the side, and then we just like put them together, multiply them together. The complicated part is figuring out how to like divide these alleles, because you have two sets, obviously. And that the way you do it is the FOIL method. If you were ever in Algebra 1 or you learned about algebra, you might have heard of it, but it stands for first, outer, inner, and last. So if we just take a look at this pair right here, I'm gonna write it out again. Okay, in order to determine the allele pairs, we would do, so it's kind of grouped like this because they're different traits. That is getting really messy. Oh well. So we do first, which is TY, capital T, capital Y, which is right here. Then outer, which is right here. Capital T, lowercase y, which is the second one over here. Right? And inner, I'm gonna do up top, lowercase t, capital Y, that's the third one. And finally, last, which is lowercase t, lowercase y. You really do not need to know this. I'm just pointing out because the hardest part about these, I guess, is figuring out how to sort them. Once you get that, it's easy because you're just like putting it together. Also, it's kind of time consuming, which is a struggle, but anyway, the phenotypic ratio for this would be 9:3 to 3:1. If you ever take a heredity test or something, they might ask you for this, and it's helpful to memorize because, again, this is time consuming, it's complicated. I don't want to go through it again, you know? I wish I drew the sticks, but you know what? Go online and search up a picture of this, because originally I had examples of like a stick that was like tall and yellow, but I got rid of them and I forgot to put them back in. So now we're here. And then we have a very long and complicated genotypic ratio. So that would be like each of these combinations. You can see that like some of these repeat, like these two right here. That would be this two right here. And it is very long, but you'll notice that it's kind of like mirrored because it is, again, two heterozygotes. So that's what happens. The main part of this was just to explain the monohybrid ones. Do not worry about the dihybrid ones. If it'll come up in like, I guess, a harder test, but for now, it's fine. Don't worry about it. Okay, and we have a practice example. I don't know how much time we have, so I might like just cut it short, but try to attempt this problem. So a heterozygous red flower with R being dominant is crossed with a homozygous white flower. White is recessive to red. So you can see that the red allele is capital R, while the white allele is a lowercase r. What is the probability that the offspring will be red? And I have the definitions down here again in case you forgot. So I'll give you guys like a minute to do this if you want to. And if you have an answer, just put it in the chat. And if no one responds, I'll just go over it after a minute or so. All right, I will wait. All right, we have one answer. Ooh, okay, another answer. Oh, so recessive just means that, I don't know how to explain. So that's where it gets complicated. So dominant just means like, I guess it's more important. I really do not know how to explain this. I came into this thinking that everyone would know what dominant recessive meant. But just know that dominant will mask recessive. So recessive is kind of like receding. How do I explain this? Okay, think of it like this. Dominant is more important than recessive. So if there is a dominant allele, it will be dominant because the dominant allele is more important. Yeah, it's hard to explain. Recessive is just like less important, I guess. Yeah, just like find a definition. Does anyone else want to attempt the problem? Because if not, I will just get on with explaining it. Okay. Oh, try not to spam. Okay, I'm just gonna move on because we don't have time left. Okay, so the first thing we do when solving this problem is trying to find out what we are crossing. So let me annotate. We know that is a heterozygous red flower. Heterozygous, we know it's going to be dominant. Well, we just know it's going to be two different alleles, one dominant and one recessive, and crossed with a homozygous white flower. So since white is recessive to red, we just have two recessive alleles right here. And then next, we draw the Punnett square. And I highlighted the red ones because that's what we're trying to find. So we put the alleles on the side. We have one here, and the other two over here. And then we just cross them together. So we put a capital R here and then a lowercase r here. And the same thing for lowercase, we just like put them together. And so we can see that two of these are red while the other two are white. Or well, they're black in this case, but the background is white, so they wouldn't show up. So the probability would then be 2/4, which equals 50%. So the answer would be 50%. Probability is 50%. So congratulations, whoever got that answer. Wait, I forgot to. Okay, sorry. So with Mendelian genetics, which is what I just talked about with like the law of dominance and wonder, there are also non-Mendelian genetics because everything is complicated. So these are exceptions to Mendel's rules. First one, incomplete dominance and codominance. So this is when neither allele is completely dominant in both cases. So for incomplete dominance, the two like phenotypes, I guess, they would mix together. So you can see we have a pink flower here, and the parents would originally be a red flower and a white flower. And this obviously goes against the law of dominance that I mentioned because I said that when you're reading together, they didn't mix, but here they do. And that is because neither allele is completely dominant. Mendel, his work was largely ignored, so he did not have a lot of resources to continue investigating. But later on, people like Hugo de Vries, they rediscovered his work, they proved that it was true, and so they kept investigating. So that's why we know this today. So incomplete dominance, think of it like incomplete. They like kind of mix together because it's incomplete, you know? So we have a blending of traits. However, for codominance, like co-president or something, that is a bad explanation, but oh well. So that is when, again, neither allele is completely dominant, but this time both alleles appear in the offspring. So you can see here, we have a pink and red flower. That doesn't look very red, but just pretend it's red. So you can see that there are white spots and there are red spots, and that's codominant. So I have a Punnett square right here, and this is kind of like an example Punnett square. This assumes that one parent is red and one parent is white, completely dominant. Well, homozygous dominant for that trait. And when you, I guess, like do the Punnett square, or when they breed together, you can see that all of the offspring have both of these alleles. So therefore, it will be either incompletely dominant or codominant. If you ever have a problem like this, they will probably tell you if it is incompletely dominant or codominant. because there's no really way to tell, because it's like literally how the organism is coded. So here I have some examples of incomplete dominance and codominance. So incomplete dominance, these are carnations, and again, these are like the pink and red flowers. They mix together, you have pink flowers. Wavy hair is also an example of incomplete dominance, because you have like straight hair and curly hair. They mix together, you get wavy hair. And then on the codominant side, we have, these are called rowan cows. It's because you have like a white cow, a brown cow, you breed them together, you have like a brown and white cow. It's kind of like splotchy. And these are, I'm pretty sure they were called Japanese 4 o'clock flowers. And you can see that it's partially pink and partially yellow, and because it's codominant, they both appear. And lastly, we have a speckled chicken, which is you take like a brown chicken, red chicken, breed them together, and it's partially brown and partially white. The next non-Mendelian genetics is multiple alleles. So this is when there are 2 or more alleles for one phenotype. And the most common example of this, not most common, but like the example you will see most often is, no, that was a speckled chicken. There was snow around it, but it was actually, maybe I took a wrong photo, but I searched up speckled chicken. Speckled chicken is a thing. It might have partially been snow, but it is a thing. So anyway, as I was saying, the most common example that you will see of multiple alleles is human blood type. So you may have heard that there are 4 blood types, A, B, AB, and O. So right here, I have the 3 alleles written out. We have A, B, and then O. For multiple alleles, sometimes it's written as like an exponent. I'm not really sure why, but you will see that sometimes. So this would be A, this is B, and O is sometimes written as just I, or I to the power of O. I don't really think it's to the power of, but that's how I read it, you know? Anyway, so this would be O. For this presentation, I'll just continue using A, B, and O because that is the simplest thing, I guess. So in this case, A and B are codominant, which is why we hear the blood type AB. Like that's why it exists. And O is recessive, which is why, first of all, that's why it's a lowercase over here. And second of all, it's why it's kind of rare, I guess. And it's recessive, okay. And over here, I have another example Punnett square. In this one, I just randomly crossed someone with AB blood type and someone with A blood type. Remember that A is dominant to O, so if I just said cross a person with A and AB, you wouldn't really know if this A person was AA or AO. But in this case, I wrote it out, it's AO, and I just did that to show you like what could happen. So first of all, in this first box, we have these 2 A together, so we just have a homozygous dominant A blood type. And for this square over here, A and B, they are codominant, so they will both appear. So this person, not this person, but that is like the AB blood type. And then over here, it is AO, which is same as this parent. And again, O is recessive, so this will just be the A blood type. And the same thing for this square, it's just the B blood type. Any questions on this? It's kind of confusing. And let's not talk about the rhesus thing. Okay, no questions. Moving on. How much time is there? I actually can't. Oh, that's good. Okay, so the last thing is polygenic, which are traits controlled by 2 or more. Oh, 1 minute? Oh, kind of. What's that? Okay. Traits controlled by 2 or more genes. So an example of this is hair, skin, and eye color. And that is kind of like the linkage thing I was talking about earlier, because they're all dependent on the melanin pigment, which you might have heard of. So if you have more melanin, you'll have a darker hair, darker skin, darker eye color. And if you have less melanin, you'll have like lighter skin color and that kind of thing. So you can see I have like a small image down here. So discontinuous would be the thing I was talking about with the Punnett square. I mentioned that it was much more complicated in real life. So it would not just be this kind of Punnett square, but for widow's peak and that kind of thing, it is just this. So dominant and recessive, it's called monogenic because it only depends on one gene. So just one gene determines whether you have a widow's peak or not. However, for skin, something like skin color, it's much more complicated, and that's because it's polygenic, and it is continuous because there are multiple genes that affect it. So in addition to melanin, there's also other things that affect like hair, skin, and eye color, but melanin is something that affects all. Okay, so I am out of time, but I am at the end of my presentation. So I have homework, which it should be posted on the website. I don't know if it is posted on the website. And it's not too difficult. I think it's optional. When I did Kid Teach Kid classes in summer last year, it was just optional because, wait, I need to go back. We can just get like points or something for it. I don't know. I guess the homework is just there if you want some extra practice with like doing things like the Kid Teach Kid website. I don't think it's been updated yet, but it should be later on. I'll just send the Google Hello. I'll just send the link in the chat, and if you want to, feel free to take a look at it and answer the questions. I guess I don't know where you're supposed to submit this. Just like hold on to it. I don't know if I'm allowed to release my email or not, so we're gonna leave it at there. And I did have a quizzes prepared, but we really don't have time. However, quizzes, I was like thinking about this. I was wondering if we wouldn't have time. So with quizzes, I can just like assign it as homework. Again, this isn't homework, it's just for fun. You could like compete with other people around here, and it's just a bunch of questions. However, with if I assign it, you can just custom deadline next week. Huh? Wait, that's not next week. This is next week. Okay, so I'll just set a random deadline. Yeah, random deadline. And I can just assign it. And if you go onto quizzes, if you go onto quizzes and enter this code, you can just like do it yourself if you feel like it. Hello? Oh, okay, there we go. Just go to quizzes. I don't know if there's a link. Oh wait, there is a link. Okay, and if you don't want to type in the code, here's a link. So the next teacher isn't here yet. So does anyone have any questions? I don't think the next teacher is here yet. Oh, yeah, question. Okay, so what is your question? Oh no, you don't have to. It's just for fun. I was originally planning to have everyone do it in class, but because we don't have time, just do it if you feel like it, if you want some extra practice, and if you're bored. I guess. It's none of this is like assigned. You're here to learn something new over the summer. It's not assigned. Any other questions? Anything about like the content? Am I in GT? What is GT? I'm sorry. Oh, could you explain what GT is? Is that like a school program? Because I don't think my school has it. Yeah, I'm from Michigan, so I don't think I have it. Just my school doesn't have it anyway. Okay. Ooh, very nice. Very cool. Anything else? If there's nothing else, it looks like the next teacher is running a bit late. Oh, thank you. The next teacher is running a bit late, so just like take a break. I guess get some water or get a snack. The next class will be forensics with Emin, I think. And I'm probably pronouncing that wrong, but I don't know the tone, so I'm just guessing. Yeah, forensics. So that would be like if you ever watched a crime show and like the detectives come in and they like analyze blood splatter and whatever. She'll be talking about that. Very interesting. Oh no, you don't need to log out. That was just a technical difficulty. The teacher should just come in, I'll make her the host, and then we'll just continue with the class. Today we had a bit of an accident and technical difficulty, so just sit tight. Go to the bathroom, get a snack, drink some water, take a break, walk around.